think they really benefit from it. I mean, I don't like to sit down with them. Yeah, we'll do that. But
Well, thank you, Michael. It gives me tremendous pleasure to introduce Ellie Armstrong. Just a little bit of background on Ellie. She actually did her undergraduate here at Berkeley, and so she's quite familiar with the place, and she worked for a while in our lab. And then she went on to do her master's at the University of Hawaii at Hilo, which is an interesting route that the reason she ended up going there was actually because she decided to go to graduate school in February. And you know, most schools have actually closed their application deadline in December, and so, you know, where could I apply in February and March? But anyway, UH Hilo were delighted to have Ellie, and she flourished there. And so she finished her master's there with Don Price, and since has gone to Stanford, and where she currently is doing her PhD with Liz Hadley and Dmitry Petrov. I should say that she did her master's and she did her undergraduate work in our lab on arthropods, and she's continuing to work quite extensively on spiders. And but as you'll hear today, she kind of went to the dark side and started working on large cats at Stanford. And this is what she'll be talking about today. She said that the central theme of her thesis is predators, and she's looking at the genomics of all predators, spiders and lions. Oh, and if you want to meet with Ellie, she has time in her schedule this afternoon. Also, we'll be going to Jupiter at 6 o'clock, so just let me know. Just email me or come at the end here and just let me know. Thanks, Ellie.
Thanks, Rosie. Yeah, so I'm super excited to be back here. A number of years ago, I was sitting on the other side of the table, kind of listening to all of the speakers pass through here, which was one of the major reasons that I decided to go to graduate school, although a bit late on that one, but made it anyways. So today I'm going to be talking a little bit about some of my PhD projects, which kind of the uniting theme, very broad theme of this, is the comparative and population genomics of the big cats. So I'll give you a little bit of an introduction for what motivated some of our questions and why we chose, or why I chose the big cats as a group to work on. And then I'll go through a couple of different projects. Some of these projects are pretty much complete, while some of these projects we're actually just finishing up data collection for, or are kind of still in process of doing data collection. So I'll show some preliminary results from those. But I would be super excited if anybody had any ideas or suggestions for how we might analyze the data, because as you will see, we are going to have a lot of data, but I think that a lot of it is super exciting and could shed some really cool insights on kind of species-wide genomics for some of these taxa. So what kind of started our group on working on some species of conservation concern? So the labs that I come from, one of them is kind of a evolutionary genomics lab. So Dimitri's group works a lot on theoretical concepts in evolution, and Liz is kind of based more on paleo type work. But they came together because both of them have been kind of focusing on what the Anthropocene looks like for species around the globe. And so this is just one of the images that I think have become super common, kind of demonstrating the shift that we've caused in taxa across space and time. So about 10,000 years ago, 99% of the biomass was represented by wild animals and 1% by humans. And this is based on calculations in 2011, and I am suspicious that this does not actually include the biomass of arthropods, but we'll get to that later. But we've then shifted that to 1% wild animals, 32% humans, and 67% livestock. So we've certainly done a number on the species that we are trying to coexist with. And kind of no matter what way you cut the cake with this, we're seeing species declines around the globe. So this is just another index which takes into account not only the diversity of the species, but as well as their abundance. And we've shown that based on this Global Living Planet Index, we've seen 50 to 60% declines, both in the number of species that we're losing altogether, as well as the number of individuals that's represented by any one species. And so the big cats are a really interesting example in all of this because they're probably the most famous and popular and well-loved species in the world, or at least some of them. So if I were to go out and just ask anybody who's walking around in Berkeley if they're on a sports team or a business, or if they use any sort of company, if you looked at their logo and their logo had an animal on it, there's a really good chance that the big cats are probably one of the animals. So despite the fact that we love these species so much, they're charismatic, we love seeing them in the zoo, they're the most popular exhibits in zoos, we're still seeing massive declines of these species in addition. So despite loving them so much, we really can't figure out how to coexist with them. So for tigers, this is just a figure from WWF. There was a decline starting about 1970 of 40,000 individuals all the way down to 3,200, which is just about the current estimate of tiger populations. I think the census that they just did put it at 3,400. And there was some goal of getting the populations back up to about 7,000 individuals by 2022, but I don't realistically see this happening. And then with lions, it's a similar story, although this is a more recent and less well-tracked decline. But we found that in 21 years, 43% of lions have gone extinct. They've been lost from 15 African range countries. And actually, this is a bit of an older number. If you look at this numbers now, it's actually, we've lost over 70% of lions across their range. So if you think about the charismatic megafauna kind of across the globe, this is not an uncommon story. So if you look at it across the different continents, with the species in black being the ones that have gone extinct and the ones in green that are still persisting, out of all of these big charismatic animals, we've really lost them from almost all places on the globe. But Africa and Asia kind of remain these strongholds where we do still have many of these species. So these are places where we kind of have an opportunity to correct how we're dealing with conservation there, what we're doing about it, how we're monitoring these species so that we don't lose them into the future. Of course, this isn't an easy task. So they face, just like all other species across the globe, they're losing their habitat really rapidly, whether it's by indirect human causes like deforestation and just habitat loss encroaching from urban areas, in addition to indirect causes of habitat loss like climate change causing sea ice to melt and leaving polar bears kind of stranded and without much of their food sources. In addition, they face another kind of interesting challenge that we've only really begun to grapple with in the last couple of years, which is the illegal wildlife trade. So these are just some figures from the globalization of crime showing that the illegal wildlife trade has been rapidly increasing and getting worse and worse over the last years. We've defined some of the routes, for example, with rhinoceros horn trading. And before, we actually thought that a lot of the illegal wildlife trade was mostly focusing on these kind of more charismatic animals, so things like rhino horn, tiger skins, tiger bone wine, things like this. But just recently, some figures have started to come out which has shown that species like amphibians, even tarantulas, the more rare ones, lizards are all being affected by the trade. So what our group wanted to focus on was how can we use kind of these novel tools to help quantify the rates of human-induced diversity loss. Specifically, I use genomics to try and answer these questions. And what I really want to understand is what are the lasting effects of all of these declines on the genome of these species. Because if we're losing diversity, there's only a certain rate with which we're going to gain it back from any population. And as we're changing the landscape, we're creating corridors that weren't there before, we're losing corridors that were there before, and we're also kind of doing these managed translocations where species that, are coming from some sort of admixed captive population are being moved across spaces to repopulate areas. How have things like the illegal trade, translocations, as I just said, and our past management decisions affected the genomic landscape of modern species? So this is particularly relevant for many African species where things like livestock fences have come up and down in a lot of places, and it's kind of halted very famous migration routes and things like that. And also, can we use next generation sequencing as a viable and informative management tool? And this is particularly important to us because normally for genetics, when you're answering a question using that tool for a very charismatic species that is, not local to North America, so it's somewhere in Africa or somewhere in Asia or somewhere on an island, the timeline of answering these questions is really long, right? So you have someone go out into a field, they get permission to collect you a sample. You then apply for permits, you wait maybe over a year, somewhere between 6 months and a year, to get the sample to a lab that has the infrastructure to do that type of analysis. You do the genetic analysis on the timeline of the scientists who are in the lab, and then sometime in the future, you write an academic paper that maybe doesn't reach everyone that it should, and you give the managers some sort of answer to the original question that they had, which is 2 or 3 years later from when they originally asked it. And that's not the timeline that conservation is operating on, or that we need to be operating on anymore. So conservation decisions are being made very quickly. Managers need to be making decisions faster than that. When we're trying to do things like convict poachers, we need the answers as to if you pull a random sample out of a bushmeat trade and you need to identify that sample, you need to know that answer now. You can't wait 2 years for a scientist to write a paper on it. So we really want to, like, our long-term goal as part of the group that I am in at Stanford is transforming these genomic tools into things that can, into platforms that can be used to answer questions rapidly. So I won't be talking too much about what we're doing with this today, because a lot of my work is kind of in this space, but we're kind of looking for ways that we can apply this, so with portable sequencing tools like the nanopore and things like that. So if anybody is interested in chatting about that, I would love to hear your opinions. So the first project that I will talk about is a project that is more or less complete, and this was really driven by Uma Ramakrishnan's lab at NCBS Bangalore in India. And what we wanted to know was, of the tiger diversity that is left globally, kind of what does it look like? What are we dealing with? So there are 6 remaining subspecies of tigers. This is a little small, but I'll kind of point these out on the map. So the Bengal tiger is native to India. The Caspian tiger was up in this region, kind of near Kazakhstan, but has now gone extinct. The Indo-Chinese tiger is in South China. The Malayan tiger is in the Malayan Peninsula. Sumatran tiger is in Sumatra. The Amur tiger is up here in the Russian Far East and kind of northern China. And so we wanted to know, what does the diversity of the remaining tigers look like? Because we only have about 3,200 tigers left. And we always say something interesting in our group is that if this were humans, we would have sequenced every single tiger in existence, right? We have far more human genomes than there are tigers. So we wanted to know kind of what are we dealing with in terms of these subspecies, with an exceptional focus on India and the Bengal tigers, because this is where really most of the remainder of the diversity is left. So we did whole genome sequencing on 65 tigers, which was an endeavor. And we found, similar to what people have gotten previously from nuclear data from microsatellites, that there are significant structure between the subspecies, which supports the taxonomic groups that have been laid out by people before. So here's the Amur tigers, the Bengals, the Malayans, and the Sumatrans. You can see that there are some kind of regions or individuals that suggest that there's some sort of historic or modern gene flow. It's hard to pinpoint just looking at an admixture plot. But that if you increase the K values, so the number of groups that these tigers can be broken into, there's actually significant structure in South India and between Central and North India. So these are really 2 more or less discrete populations. And if you look at the FST values, they actually are about as different from one another as any of the subspecies are from each other, which is really interesting. So the next thing that we wanted to do was kind of to address this misnomer that's been happening in the genomics literature, where somebody takes a single genome and they calculate a heterozygosity value, and then they put it on a plot with a bunch of really fun other endangered species and maybe humans, and then they say something about the risk of endangerment of that species. So we looked across the 65 tigers that we had generated genomes for and calculated the heterozygous number of SNPs per megabase, and found that there was substantial variation both within populations as well as between species. And so you can see that if you just are using a single genome to make a heterozygosity estimate, you're really not getting the whole picture of what's going on at the species. So although these plots are really fun and it's really nice to throw an endangered species up and say, oh, it's more in red than the Tasmanian tiger, it might not be super informative in terms of what is actually a species of conservation concern. And to break that down a little bit more, we also looked at the runs of homozygosity between and within populations. And so what this is doing is it's trying to quantify the amount of inbreeding that has occurred in both recent time and that is perhaps a little bit older. So the idea being that short runs of homozygosity, so runs of homozygosity that are in bins between 10 and 100 kb, indicate older inbreeding or population bottlenecks where related individuals are breeding with one another. And then larger runs of homozygosity indicate recent inbreeding. And so what we found was that for the short runs, all of the species seem to have more or less a pretty even amount of these bins of runs of homozygosity across their genome. But when you get into the larger bins, you start seeing more substantial differentiation. I want to point out this individual. So this isn't a Moor tiger. This is actually the original tiger reference genome. So if you go onto NCBI right now and you search tiger and you download the reference genome, this is the individual that you'll get. It's supposed to be an Amur tiger. It was collected from a Korean zoo. However, as you can see, it has very, very long runs of homozygosity in the genome, indicating that it has probably been in captivity and inbred for a long time. And if we add it into our admixture plots, we actually detect some gene flow from it into other subspecies. So I think that's another important reason that when you're using specimens, whether you don't know where they're from because they're from a museum and they haven't been geo-referenced, or if you're using a captive individual from a zoo or from a sanctuary, that it's important to consider whether or not it's representative of the individuals that are in the wild that you're trying to say something about. And then also just to point out, so here are the Bengal tigers. So again, we found that there were varying levels of runs of homozygosity across these genomes, indicating that there was some sort of evolutionary split. And then if you look at the total amount of the genome in ROH, it's variable across individuals, but they all roughly have the same mean levels, with some individuals kind of being these outliers. So what does that actually mean for tigers, and what can we say about them in terms of their demographic history? So some of our collaborators, Laurent Escoffier and several of his students, did a really awesome demographic analysis for us. And they estimated that the subspecies divergence, so if you imagine this to be a really large metapopulation of all tigers before the subspecies began to split, we estimate that they diverged in the Holocene, or around the Holocene, almost 10,000 years ago. So this is in stark contrast to every estimate that has been made before. So estimates previously put the subspecies divergence at around 100,000 years ago. And many of these papers, even the ones that did look at whole genomes of tigers, did not actually include individuals from the Bengal subspecies and had very sparse sampling of some of the other subpopulations, which means that it could have kind of biased the estimate and not made available kind of these models of these metapopulations. And then in addition, with the Bengal tigers, because as I said, what we were really interested in is India, because most of the diversity of tigers most of the diversity of modern tigers reside in India. So kind of a bulk of our tiger diversity, a bulk of the individuals that we have left are all within India. And we wanted to know were the splits that happen between these populations in India. So as I showed before, we kind of have this central and north split in India with the south. We wanted to know, was this something that happened really, really recently in evolutionary history? Like, is this something that happened within the last 100 years? Urbanization starts really kicking up and the populations can no longer exchange genes with each other? Or was this something that was kind of a more historic event? And it looks like it was actually a little bit older, so it's 2,000 years ago, definitely still coinciding with the time of bursts in human activity, but it was not so recent that it's something that we really drove and really recent management decisions have led to. We also detected that there were very few migrations after the initial divergence, indicating that really from this large metapopulation of tigers, perhaps they kind of just spread out in a burst. And this is not entirely unexpected, because what we can tell from the history of big cats and Panthera in general is that the speciation seemed to happen really, really rapidly. So kind of all of the different species within Panthera may have occurred more simultaneously, which is why a lot of the time we detect these signals of hybridization, possible hybridization, or introgression across the genomes. And last but certainly not least, we wanted to look at selection to see whether any of the populations had any kind of adaptive differences between them. Because of course, if you're making decisions about how you're going to be managing tigers into the future, knowing whether there's any unique genetic variation that may confer some sort of fitness advantage across the populations is important. And so again, this was led by Laurent Escoffier's lab. They looked at the MPBS statistic, so just a modified population branch statistic. They did some very fancy math, which I will not explain to you here, but you can check out our preprint, which is on bioRxiv if you're super interested. And what we found was that there were really two regions that kind of popped out to us that the genes were annotated and we could say something important about them. So one of them was, we found that there was signals for strong selection in the Amur tigers for adaptation to cold tolerance. So if you remember back from the first slide, the Amur tigers are from the Russian Far East and China, so this is really the northernmost part of the tiger range. So a lot of this makes sense. They're dealing with colder conditions year-round in general, and also going up to higher latitudes. In addition, we also found that there was some selection in the Sumatran tigers that may be related to body size. So this is a really interesting one, and I'm not sure how much, I guess I shouldn't say how much I believe it, but it's an interesting thing to think about because tigers are exceptionally difficult to tell apart. So if I set a tiger in front of you, or even if it was a person that was an expert, a world expert from a museum that knew so much about tigers, it would be nearly impossible for them to tell you what subspecies of tiger they were looking at. They are so phenotypically similar and have such broad phenotypic distributions across their morphological traits that it's very difficult. In fact, the cats are so difficult to tell apart, skeletally especially, that it's even challenging to tell the difference between tiger and lion skeletons in a lot of cases, which is why lion skeletons are sold as tiger skeletons on the black market and nobody can tell the difference. Sumatran tigers are said to be slightly smaller than the other tigers. Whether or not I believe that's true, because I think that they're phenotypically pretty broad, but the genome says otherwise, so it could be the case. So in conclusion with that project, we did find that there was significant structure between the subspecies, which reinforces the taxonomic kind of separation among them, and this is something that has been extremely highly debated. So I think the tiger subspecies have been rearranged like maybe 15 times in the last like 20 or 30 years. Like, what is a subspecies? What is not a subspecies? How many taxonomic units there are? But it also was super important for us to know that there was significant structure between central and north and south India. So kind of the split down the mid-center of India, splitting central and north from south. So this is something that UMA is actually putting into management action now. So they were planning on migrating tigers between these locations, but they are no longer going to do this because we've showed that they're significantly differentiated, and if you merge these populations, you then homogenize the amount of diversity and then possibly lose diversity over time, which is not what we want with tigers. In addition, we found that heterozygosity values, if you're using this as a representation of how endangered a species is of going extinct or being perturbed by disease or disasters, etc., varied substantially across subspecies and individuals. So this is just a cautionary tale of one genome does not equal the species level heterozygosity, and you can't use a single individual to kind of represent any population. We showed that runs of homozygosity varied across subspecies, but all of them showed signs of historic and some recent inbreeding. So this was super interesting to us, and again, with these ROH categories, Uma is following up on this work with one of her students who is looking at one specific population in Ranthambore who were kind of worried about whether or not they were actually starting to have some inbreeding depression, so they started showing some of the morphological signs of having inbreeding depression, and some of our highest ranked ROH individuals from the Bengal data that we had here were from that population, so that's something that they need to monitor more intensely, and so they're going through right now and verifying all the pedigrees and the parent-offspring pairs. In addition, we showed that selection showed possible adaptation to cold tolerance in the Amur tigers and body size in the Sumatran tigers. And divergence is relatively recent compared to what the estimates that we had before. So 2,000 years for the different Bengal populations and 10,000 years for the different tiger subspecies. So this is a very kind of rapid and reactionary change for the cats. Okay, so now I'm going to shift gears a little bit into a second project. So we just finished data collection on this project. This is still about tigers, so I'll start and stay on tigers for a little bit and then switch to the more social sister species. So captive tigers are this has been a really interesting project, sort of to go off on a little bit of a tangent about what happens when you're doing a PhD and how much unexpected things you run into. So we got involved with this because while we were collecting tiger samples from around the country, so we were asking people like the Frozen Zoo, we were asking museums, we came across this organization that told us, yes, we can get you hundreds of tiger samples. And we said, what? Where do you have hundreds of tiger samples? How do you have hundreds of tiger samples? And they said, well, they're captives. And we said, what do you mean captives? Like zoo captives? And they said, no, they're from sanctuaries. And so we were like, huh, okay, that's really interesting. What is, like, you know, we know what an animal sanctuary is, but like, where did these tigers come from? Like, are they from the zoo? Are they from, like, how did these people get these tigers, right? And so then they tell us that there's somewhere between 5,000 to 10,000 of these individuals in these facilities in the US. And we were like, holy, that's more tigers than there are in the wild in captivity in the US. And so when we started looking into this issue and talking more with this organization, it became very apparent to us that these were not good representative individuals to look at wild tiger kind of demographic patterns and history and evolution and how we might preserve those species, but it was more of an illegal wildlife trade and captivity type project. So America has a tiger problem. So like I said, these are individuals that are in non-accredited sanctuaries, backyards, private zoos, roadside zoos, places that are kind of calling themselves sanctuaries but are actually breeding facilities. So we have many, many, many tiger farms in the US. So if you kind of go online and you see, you know, those videos of the dodo flying over these facilities in China and Thailand saying like, oh, they're breeding hundreds of tigers here and selling them for tiger bone wine. It's happening here too, which is really alarming, right? So this is happening and it's like, as a biologist, you're like, how did I miss this? Like, how do we not know that we have tiger farms in the United States? So why are there so many captive tigers in the United States? So it's a money-making industry. So historically, a lot of this was driven by the circus. So a lot of the reason that the rules and regulations for owning a captive tiger are lax because when the circus was still going on, so this is like Ringling Brothers, Barnum and Bailey, they were strong lobbyers for there to be really no rules and regulations for owning cats, right? Because they wanted to do whatever you want. So if you're a circus owner, you need to be able to keep animals in small cages. You need to be able to transport them easily across state lines. And you need to be able to breed them because you need a constant supply of tigers. And this is, of course, not only happening with tigers, but it just so happens and weirdly turns out that tigers behave and mate really well in captivity, which is not good for them, it turns out. But the other way that these people are making money, because, so like I said, the circus closed down about 4 years ago. I don't know how common knowledge that is, but there's no more of these kind of big circuses in the US. So what these other people are doing is this, for the most part. So if you show up to one of these places, it'll look very similar to like a really great sanctuary. You'll go in, there will be animals just like you would see in a zoo, and you'll go see the tigers and the bears and yay. And then you'll get to the back and they'll say, okay, do you want to pay me $50 to hold a tiger cub? And you know, as a person who has no idea, you're like, oh my gosh, I want to hold a tiger cub. Like, this sounds great, right? Not great. So there are also, in more rare instances, so this is actually a photo from Thailand, there are more rare instances where they will sedate adult tigers to the point where they, you know, couldn't really do anything to you, so that you can go in with them. But after looking through all the data, the question that we had was, well, if the clutch size of a tiger is between 3 and 5 when they first start breeding, of course you can send the female tiger back into heat if you take her cubs away right away, so they can produce quite a few cubs in a year if you force them to. The question for us was, okay, so there are a lot of baby tigers, right? But there are not as many adults as we would expect for how many baby tigers are being produced. And as soon as you reach like between 6 to 8 months with a cub, they are now this kind of like very difficult to deal with eating a ton of meat a day. Like, they're very costly animals, and you no longer can go in with them. They're dangerous. And so they're clearly not letting all of the adults survive. So what is happening to the cubs that are turning into adults that are not kind of being circled back around into this like breeding scheme? And we were very suspicious that probably what is happening is something like this, something like this. This is a tiger that just shows all the different parts of the body that are used in traditional Asian medicine. So if I am someone that has sold tiger skins or bones or whatever it is that I'm selling, why would I risk shipping a tiger skin over an international border if I could just get it from a seller in the US? You're like barely risking anything, right? And the person that you're selling it to can't tell what the tiger is. Like, you could tell them this is a Royal Bengal tiger, and I don't think that there's any person on the planet that would go, no, this is not, this is a mutt tiger from a sanctuary. Like, you can't tell, right? Like, it's completely, you can't tell. So we were suspicious that a lot of these individuals were winding up probably in repositories in the US Fish and Wildlife Service where they put these confiscated products, or ending up in these kind of more breeding schemes. So if we break this down by the numbers, see, we kind of just were referring to them as backyard tigers for lack of a better term. So these are the places that are these non-accredited sanctuaries that are breeding the cats, that are letting them be held for cub petting, et cetera, et cetera. We have 3,400 to 3,800 in the wild. We have 1,000 in the accredited sanctuaries. So when I say accredited, what I mean is there's no breeding and there's no contact with the cats. And then we have about 300 tigers that are managed in AZA zoos in the US that represent 3 subspecies. So we have an Amur breeding group, we have a Sumatran breeding group, and we have a Malayan breeding group. And I took this picture. And that's probably my peak photography moment, so enjoy it. So we wanted to know, first of all, probably this is kind of what is happening. Historically, maybe some individuals had come from a zoo back before the AZA was super organized and was still selling to private breeders and things like that. But we wanted to know, okay, if we sample this population, because when these places get shut down, we rehome them here. So we wanted to know, okay, but where are these individuals coming from? Like, are they coming from the wild? Are they coming from the zoo? We have no idea. We don't even know what they are. They could be pure subspecies. We have no idea. So we started doing genome sequencing, which is pretty much our default when we don't know what's going on. So this is just kind of some of our early data. So we now have, I think, 150 individuals from these different captive facilities across the US. And so what we found, and these are just, I think, 5 individuals each from the representative from the paper that I showed before. And here are our captive tigers. So you can see that they're kind of all over the place. What's the first interesting thing to point out is I told you that we only have 3 breeding programs in AZA zoos, which means that these guys, so the Bengal tigers, probably are spillover from the circus. So if you remember back when Ringling, if you went to the circus when you were a kid, a lot of the time they have white tigers. So the white tiger phenotype is native to the Bengal tiger. So it is a wild phenotype. It occurs naturally in the wild, albeit extremely rarely. And it's only ever been observed in Bengals. And we know that they had brought over some white tigers. At one point there was a zoo breeding program, but there's not anymore. And the circuses were trying to breed a lot of white tigers for their shows. And so we were really surprised to see that although the white tigers do have some Bengal ancestry, they also have a lot of other ancestry, despite the fact that they're still white. So these people have pretty much found a way to still get the white phenotype, but breed between the subspecies. And so what we're doing with this data now is we are going through and we're trying to understand the relationships between the individuals, because like I said, these are kind of mass breeding programs. And if you're a tiger farm and you're breeding lots of tigers, then you kind of sell them out to a burst of sellers. So we have a lot of tigers coming in from different places that are probably related to one another, siblings, parent, offspring, things like that. But they don't know it because they're getting them from different facilities because they've been sold to 2 different places. So we're going through and we're trying to reconstruct pedigrees for these guys, in addition to doing some chromosome painting to see what portions of the genome kind of are coming from each of the subspecies ancestry. And then the last thing that we want to do is we have some confiscated specimens from the U.S. Fish and Wildlife that we are going to try our very best to get DNA out of and see what proportion of these are actually from mixed tigers. Because we suspect that the proportion is probably quite high. In addition, I have an undergraduate who worked with me this summer from Duke, and she's holding a penguin here because she's the luckiest person in the world. She was able to do some maternal line tracing and verification. So cats are the most difficult thing in the world and have pneumites, so nuclear mitochondrial inserts, all throughout their genome. So when you do whole genome sequencing of cats, it's almost impossible to pull out just the mitochondrial genome so that we could trace some of these maternal lines, especially for the white tigers. Because it's really interesting for us to know, like, were people shipping white tigers over after the original founders came over? Because there's some talk that there was only a single founder for white tigers in the US. And we'd like to verify that if we can. So she developed a method. Oh, okay. It's just appearing randomly. Yay, okay. So she developed a method to cheaply and quickly exclude nuclear DNA from extraction. So you can do this in a lot of different ways. Like traditional methods was to run a gel, it runs in both directions. So you separate the linear DNA from the circular DNA because your mitochondrial DNA is circular, so then you're not sequencing the pneumite. Because if we just use primers, right, we're going to amplify the pneumite because it looks exactly the same as the mitochondria except for a few base pairs of difference from the insertion. So she looked into two methods using DNA dilutions as well as linear DNA degradation. The DNA dilutions did not work at all. We could do a 1 to 100,000th dilution and we would still amplify the pneumite, which was kind of incredible. And then we tried a linear DNA degradation. It's just an enzyme, so it's really cheap and really quick, and that was kind of our best case scenario. So then we were able to get clean reads from the mitochondria, which was really nice. And we have tried to do this bioinformatically a number of times with the next-gen data because you do get a lot of mitochondrial reads, right, if you're just trying to amplify the whole genome. But unfortunately, it's really inconsistent and basically doesn't work at all. It's almost impossible to tease apart because there are so many insertions into the nuclear genome that you can't figure it out. If anyone has, let me know. Okay. Sorry. Okay. So what we're doing next with this is that we're hoping to do some PacBio sequencing of the mitogenome. So now that we've kind of isolated the DNA that's just from the mitogenomic reads. And then we'd really like to do nanopore on some of the actual tiger genomes to figure out where in the genome they have been inserted. So based on the genomes that we have so far that we've done de novo assemblies for, if I just blast for the pneumite, so there is a published pneumite sequence in NCBI from an older paper. If I just blast for that in my genome and try and find which scaffold it hits to, I get a hit on every single scaffold, which is not ideal. It could be inserted on every single scaffold, or on every single chromosome, we don't know. But we'd like to kind of narrow down the location of these, how much expansion there has been. And this is actually also a really exciting thing in terms of phylogenetics in general, because if you can date these pneumite insertions, you actually can start getting better dates for the splits between the various members of Panthera, in addition to Felidae. Oh, again. There we go. Okay. Okay, so now I'm going to switch gears to lions. So this is the kind of the bigger project of my PhD that I've been working on for the past 3 years. So we wanted to do something similar with lions to what we did in tigers and sort of do a species-wide sampling using genomics to see what kind of variation we still have remaining. So just to kind of cover a little bit more about lions, they are listed as vulnerable. They've disappeared now. This says 12, but it's 15 countries in the last several decades. And they really only have a few remaining strongholds. So 3/5 of the largest populations are all located in Tanzania. So places where they used to be, they just are not anymore. So they're extremely rare in West Africa, despite being relatively common at the turn of the century. And in South Africa, we have this really weird situation where they do have some strongholds, for example, in Kruger National Park, but for the most part, these are admixed and managed populations that are completely cut off from other populations. So it's kind of these groups of people that have private land, that have groups of lions, that occasionally trade lions with one another. And what's interesting about these lions is that when some park that's up here says, oh, our lions are getting a little low, we could really use some more, these are the ones that they move up. So there's been some weird genetic things that have happened. Not all of it has been super well tracked. And so an easy way to kind of figure out what's going on there is to use genomics. So the first thing that we did is we built a lion genome assembly. We used a female individual from one of our sanctuaries. So these are the same places where the unfortunate tigers end up. So it was a female lion from the Exotic Feline Rescue Center, and she has a very sordid history, so we wanted to use her as an ambassador animal, which is, it's not a wild lion, but you know, the chromosomes all look the same, so we're lucky with that. It's not like with rodents where there's lots of rearrangements and things. The felid karyotype is very stable. We used a variety of technologies, including 10x, dovetail high C, and nanopore for gap filling, and we got really good statistics. So we basically have chromosomes. It can obviously always be improved, as all genomes can. But we have a scaffold L90 of 18, and there are 19 chromosome pairs in the felids. And this is just an alignment with the domestic cat. So there are some rearrangements. Some of them may be noise and kind of regions of low complexity that are just mapping in two places. So again, the first thing we did was looked at heterozygosity. So what we wanted to show here was that, again, for these kind of estimates of heterozygosity that people make, what we wanted to show is that it does make a difference what you're using as your map to reference, and that when people are pulling these statistics from one paper, this is sort of like a microsatellite issue, right? So like you can't compare microsatellites that are run between two different labs unless you have a control set. And we showed pretty much the same thing. So if you map an Amur leopard to the Amur leopard genome, you get a different heterozygosity estimate, a vastly different heterozygosity estimate than if you map it to the domestic cat genome. And this is pretty much consistent across all of these felid species. It was especially apparent in the Asiatic lion. So the Asiatic lion is basically hanging on by a thread in terms of as far as species go. It's extremely homozygous as far as we can tell. There's very few individuals left, I think less than 200, and they had bottlenecked down to maybe less than 100 individuals a couple years ago. But you can see that pretty consistently you get a higher and possibly more accurate estimation of heterozygosity if you map the genome, the species to its own genome than if you map it to the domestic cat. So this is important, especially if you're looking at the patterns of heterozygosity across the genome. In addition, if you're doing these relative measures. Just a note, in case anyone is interested, this also differs very much if you're using a different method or a different program of estimation. So a lot of the time people will grab these heterozygosity values from other papers and kind of just compare them with whatever they estimated. But if you're not using the same software, the same version, you're going to get two different things. This is kind of no matter what way you cut the cake. So we also looked at the patterns of heterozygosity across the genome. So this is just the binned het estimates across the different chromosomes. So these are 4 different lions. So this is the lion that we did for the genome assembly. So she's pretty diverse. This is probably because she's from a more mixed stock. So we don't know her background. Hopefully when we have more data, we'll be able to pinpoint kind of where the different parts of her genome are from. These are 2 lions from a paper that looked at felid phylogenetics published in 2013. This is a tawny lion, and this is actually a white lion, so people do breed white lions, which is kind of interesting. And then this is the Asiatic lion, which is just an alarming graph to look at. So this thing barely has any heterozygosity whatsoever, which is, you know, maybe heterozygosity is not the best indicator of whether or not a species will survive and persist, but this is just a little much. So we also looked at demographic history. I'm just going to speed up a little bit here so I can get you guys out on time. We found some interesting things with demographic history. Most of the lions shared similar histories. The Asiatic lion did have this kind of up here, and then our genome also had this increase at the end. And PSMC graphs, for those of you who are familiar with them, they use heterozygosity estimates kind of in these slides across the genome to estimate past coalescent times for the different parts of the genome. And what we think is happening is that probably the Asiatic lion is losing signal here because there's no longer any heterozygous sites for it to figure out what the demographic history is. And possibly because our lion is more heterozygous, we're losing signal here. So there are better ways to look at demographic history if you have population level data. So we're hoping to do that once we have more genomes. Just gonna speed up. Okay, so this is kind of the meat and potatoes, but I've spent the last 2.5 years collecting over 700 lion samples from over 50 localities across Africa, in addition to Asia, so India where the Asiatic lion is. These include modern and museum specimens. We have over 20 collaborators in range countries. And so we're really excited to start digging into this data and seeing what's going on with the lion. We do have some preliminary results from Benin, Zambia, Botswana, Botswana, Zambia, and then Uganda. So what we see so far is that what we found was super interesting is that just based on these different places, so Okavango West is going to be this in Botswana, so it's kind of West Delta within Botswana. Zambia is right next to Botswana, so it makes sense that these are clustered at K2. But Chobe, which is in the west of, is in the southwest of Botswana, has kind of this shared history. And if we just are plotting it with Benin, it's actually part of its genome is actually clustering with them, which is super interesting and kind of goes to show that if you are sampling these species that have really, really wide ranges, if you're missing a sampling locality, you're probably going to get kind of an odd answer based on your structure plot. And then I'm going to rush through this stuff. So we also have kind of some case studies, one of which is from the Ngorongoro crater in Tanzania, which is here. So we have a kind of series time point of data. There's just some really gratuitous photos of the crater. Visit there if you can, it's really beautiful. So the Ngorongoro crater lion population is very insulated. There's been hardly any migrants in and out of the population. It's a very small lion population, and it's gone through a number of extreme bottlenecks. So one was in the early 1800s from a rinderpest outbreak, which affected the ungulates in the area and basically depleted their prey source. And then there's been 2 canine distemper outbreaks since then. And so this population has been extensively monitored, starting in about, it was actually a little bit before this. So in the 1950s, they started kind of tracking all the individuals in the crater because there were so few of them, they could make these really nice pedigrees. But like I said, there were these really horrible canine distemper outbreaks in the crater. This not only affected lions, it also affected hyenas and other carnivores. And so we were lucky enough to get 2 sampling periods from kind of around these outbreaks from a couple of different sources. And so what we'd like to do is look at kind of the rebounds of diversity across time and how it's declined based on the outbreaks, see if it's affected any of the regions in the genome which are responsible for immune function and things like that. We've had pretty good luck with it so far. Like I said, we also have some hyenas from the crater, but if anybody has any tips and tricks on how to get really good DNA out of very old plasma and serum, I would be all ears. So we're trying this kit right now, which is a new, someone made an ancient DNA kit, which I think is really nice for people who are maybe not necessarily set up for ancient DNA, but would like to do something and also grab all of the single strand DNA pieces from their extraction. And then last, I'm working with the Smithsonian Conservation Biology Institute, so with Rob, who will be here next week, as well as Jesus Maldonado and Michael Campana. So we have 59 lions that were collected, shot, and sent back to the Smithsonian by Teddy Roosevelt from Kenya. So this is a really incredible data set, and we've been able to get quite a few really good genomes out of them. So of the 59 samples, although some of them we've barely been able to amplify anything, 10 have genome coverage over 50%, and I think 7 of those were upwards of 90%. So we've managed to amplify the whole genome from 120-year-old specimens. Of course, since I've never worked on ancient DNA, I was like, oh, these don't look very good. And they were like, what? These look great. And so anyway, so we are very lucky. We'll also be trying to estimate directly the decline, the diversity decline between these historic specimens and the modern specimens that we have from Kenya. And we've also shown that there's very little DNA damage. From just a few of the lions that we had genotyped of these historic samples, we showed And this is these plots have hardly been corrected really, or these reads have hardly been corrected at all, so this could change a little bit, but we think that it's a pretty good first kind of estimate. So here are 3 historic lions. These are some modern lions from Botswana in blue. These are 3 modern captive lions, and then this one is the Asiatic lion, as you can probably tell because it's at the bottom of the plot with no diversity in its genome. If you know anyone who has more lion samples, historic or otherwise, in addition to hyenas and African leopards, we would very much like to collaborate with you. We're definitely looking for more samples because the Roosevelt collection has a number of other specimens that they would like to estimate the diversity climbs in. And I have a million people to thank because in order to collect 700 lion samples across so many countries, it takes a whole lot of people saying yes to you, even though you're some crazy genomicist from California saying, we're going to sequence all of your samples, and they're like, what? So I'm really, really grateful to everyone who has kind of gone out on a limb for us and given us the samples, trusted us with the samples, as well as to our funding sources, which includes a really generous donation from the Wood Tiger Fund, Tigers in America, which is the sanctuary kind of network I work with, the Snow Leopard Trust, and my funding from the GRFP. I'm sorry that this last part was a little bit rushed, but I wanted to tell you guys about all the cool samples that we have. So thank you for listening, and thank you for having me, and I will take questions.
So we'll let people who need to run to class get out, and I'm sure Ellie will be happy to take questions.
Yes?
I was wondering how you calibrated your estimate of the dates. It seems so recent for potentially some big differences between populations.
For the tigers? So this is actually kind of an interesting tangent, which I didn't get into here. So there are 2 estimates actually for felid/tiger mutation rates, which I think could be driving some of the differences. And we had to do a little bit of sleuthing to understand in the literature whether this was like a double division error by someone, but there are 2 estimates and they're an order of magnitude off. So like one is 10 to the minus 8 and one is 10 to the minus 9, right? Which would kind of put you in that, are you 10,000 years or are you 100,000 years, right? It kind of explains that. But we actually use the same estimation that the paper that got 100,000 years used. And so in terms of the data that we used to kind of correct out, so we excluded exons, so regions of really high diversity, things like that. So we kind of went through all of the parsing that we could have gone through from the genomic data, and we only used individuals that had greater than 10x coverage, so we were really confident when there were differences between sites. But I definitely think it's worth, you know, when you're doing these demographic histories to take whatever mutation rate estimate that you're using with a grain of salt, because everyone's kind of using this generic one for mammals, right? So we actually have another kind of tangential within project where we're trying to estimate mutation rates using pedigree, because we have obviously some pedigrees within our data that have just occurred naturally. So we're trying to estimate some mutation rates for them. Hopefully it's not too much different than the one that we used, but I mean, I think that would be interesting anyways, just to know what it actually is. Yeah, I hope that answered your question. Yes?
So in your captive plots, you had quite a few that showed up, like the Sumatran and also Malaysian, and which was interesting because in theory the captive one, I mean, do you think those are illegally collected animals that were then sort of infiltrated into those, or from zoos or circuses, or where did those sort of rare tigers come from?
So it's really hard to say. So our best guess is that before AZA, so the Association of Zoos and Aquariums, existed, or when they were in their early days, there was a lot of buying and selling between the zoos and private owners. So they were working with a circus to buy tigers, trading tigers back and forth, things like that, even though they were maintaining their lines in a pure way. So we think they could have come from there. But honestly, it's really hard to say. So we're trying to figure out exactly what happened. Part of this is going to be going through like CITES approval records to see when people have shipped tigers into the country that maybe went to private owners, because they could very well be from different wild stocks. And another reason that we're really excited about understanding that question exactly is that this population of captive tigers is much, much larger than any of the zoo populations, the wild populations. And so there's a very good chance that there is diversity that is now extinct in the wild that is still in these populations. So in terms of like conservation value, there actually might be a ton of conservation value in these cats. Even if you ask like, you know, a traditional conservation person that works on the ground with tigers, they'll say, oh, you know, we don't want anything to do with the captives. But as we're moving kind of more into this era where, you know, people are doing in vitro fertilization in rhinos and things like that, and we're having to consider some of these options, I think it's just nice to know what tools you have in your toolkit, right? You have the whole picture. So yeah, but that's a good question. I don't know. I hope we can figure it out. Yes.
You said that the subspecies were phenotypically very similar. It's hard to tell them. What was the basis for the original designation of those subspecies?
So the original one, I am not sure. So I know that there is a paper that used both nuclear and mitochondrial data in addition to things like, I think a lot of it has to do with it. I'm not a taxonomist, so I wish I could answer this question better, but I believe that a lot of it was kind of jaw measurements. There was some sort of pelvis kind of angle that they could use to make the subspecies distinctions. But yeah, it's apparently extremely difficult, and a lot of the original specimens that they had used as kind of the representative for any of these given species have been kind of strongly disputed. So even for the sites like in Tanzania where they're digging up really, really old leopards and lions, they're having trouble. There's like fights over whether it's a leopard or whether it's this or whether it's this. So it's, I don't know. Yeah. Sorry, that probably didn't answer the question very well, but I think it's really hard with cats from what I understand.
Jim, you look like you want to say something.
Well, no, I just, you can assume that those, the tiger subspecies were probably from, you know, the British Raj, and so they date from the 1840s, something like that, and so the basis for it is mostly probably geography more than anything else.
Well, that's what I was...
But whether or not they've ever been, you know, whole samples have been examined to find morphometric or other kinds of traits that fit down the geography, I have no idea.
Yeah, I know that there was a time period where there were many, many more subspecies than 6.
Yeah.
And then just recently, based on kind of like within the last 10 years is what I mean by recently, it went down to, I think only 3 was what the kind of CITES designation was for a while. And then just now, in the past like 3 to 5 years, they've gone back up to 6. So I think that it's one of those groups, canids and felids, I think get rearranged pretty frequently. And we, you know, only just like within the last 5 to 10 years, we decided that the snow leopard is actually the sister species to the tiger. So I think it's pretty open for discussion in those terms. Yes.
I was going to say, sometimes with some of these really endangered species, it's political, because
Yes.
you can say that you have only representative individuals in your country or in your park from a subspecies, then you can actually make a case for people to, you know, for saving them. So often, I don't know about tigers, but with apes, that's often.
That is absolutely the case. Yes, you are very right. And it is, tigers are probably the most political species that I can think of. So both groups in China and groups in India have a very strong feeling about where it should be said that tigers originated. So that makes doing this kind of work even more sensitive and political, and, you know, so there's a reason that we were unable to get, you know, Indo-Chinese and South China tiger samples, even though we really would have loved to include them, but they're just, you know, it's virtually impossible when you're working with Indian researchers to get access to some of these collections. So hopefully as data becomes more public, we can add to our understanding of such things. So. Yeah, sure.
So I was just wondering, Ellie, kind of building up on the time idea and exactly how many species are there. You said there'd been a big burst of diversification, whatever, 10,000 years ago, but relatively recently.
Mm-hmm.
In response to what? What was changing in the world? Do you know when they
So
diversified?
this is a really good question for Liz, because she knows this stuff much better than me. But from what I understand, so I have actually looked into this a lot more in terms of the diversification of Panthera as a group. So they think that a lot of this possibly corresponded with the Himalayas kind of making this massive transition. And then in terms of the tiger subspecies, for the Sumatrans, the divergence time that we estimate actually does correlate with sea level dropping, I believe, around the time. So the Sumatran Peninsula is basically, becoming inhabitable. So some of our dates correlate with those kind of geologic estimates. But I think in general, the Himalayas being kind of this spot of diversification, there's been some suggestion in other groups, like in pikas, that it drove this big massive burst in pika radiations, and as well as other groups. But I think geologically there's a whole lot of debate about that in general, so it's hard for us to pinpoint an exact kind of event that made this happen. Yeah. And there's so few specimens too. So that's another really hard thing about working with cats, is that there's maybe like 5 or 6 fossils that we're very sure are what they say they are. So it's very rare to be able to calibrate these dates in a meaningful way. Yeah.
Maybe one last question?
Sure. With the circus no longer able to use these animals, do you think we're at a point when the laws or the enforcement of existing laws might change? And if so, what might happen with the 5,000 to 10,000 animals?
Yeah, that's a good question. So the group that I work with, a lot of what they do behind the scenes, and I'm not as involved with, is trying to get these laws into action, because the circus is no longer lobbying against anything like this being passed. Quite a few of the facilities have started to get shut down by U.S. Fish and Wildlife Service and USDA. There's a big Cat Safety Act right now that we're hoping to get into Congress. So if anybody wants to go sign their name on a another ballot measure, which I feel like we're doing that more and more now, that we're hoping that will get passed, and that will shut down a lot of these places. Because right now, so for example, just like some of the harrowing stories is like, we have rescued pairs of lions that have been chained in farmhouses and have never walked, so their back legs are completely useless. We rescued leopards that were being kept in oil drums. So there's just, we have not enough space in all of the kind of accredited sanctuaries that we work with for all these animals. So I think that if there were some sort of kind of big push to get all of these animals out of these facilities, the zoos would need to be more responsive to housing these animals, because right now zoos do not want what we call these generic tigers. They don't want mixed tigers. So there was a cub, maybe you guys heard about it, last year that got smuggled across the U.S.-Mexico border. The rapper Tyga was smuggling it across for himself, which is a little bit poetic in a way. But it got confiscated at the border, and it got sent to San Diego Zoo, and they asked us to sequence its genome because they wanted to know what it was, because they had another tiger cub in their facility. And so we sequenced it, and it came back as being completely mixed. And so we said, it's mixed. But then we were like, oh, this will be great. He'll have a playmate. Zoo kicked it. They didn't want it. So they say that they don't have space in their facilities for any generic tigers. The exception to that is your local Oakland Zoo, who actually all their tigers are generic tigers. So we very much appreciate that they are willing to house some of them. But yeah, it's a pretty nightmarish situation, I think. If this were kind of to just all break down at once, there would be not many places for them to go. Yeah.
Okay, and Rosie, did you want to remind people about?
So yes, if you could just if you could email me or let Carly know if you'd like to meet with Ellie, if you'd like to set up an appointment.